Hey there. The purpose of this segment on proteins, it will be part 3, is to talk a little bit more about chirality. I realized that in the last presentation I did not talk about chirality as much as I would have liked to, so this one is focusing just on this concept of molecules having mirror images, having different isomers that are mirror images of one another. So with this, I will pop into my PowerPoint. I have to share my screen here. And I will start the show. So there is this concept of handedness. So, and in my presentation before, I talked about how to put your hands up next to one another and see that they are mirror images of one another. So mirror images of hands will not superimpose on each other, but your hands are mirror images of one another, and they will not superimpose on each other. So if I have each of the backs of my hands facing you, you can see that they're not the same. One thumb is coming out this way and one thumb is coming out this way. So when objects have this characteristic, they're considered to be chiral. Now, when we're talking about carbon-based compounds, what we're really looking at is a carbon. In order for a carbon to have chirality, there have to be 4 different groups attached to it. So if we look at this, this is the carbon. We call this the chiral carbon. And one of the groups attached to it is this carboxylic acid group, and another is a hydrogen, and we have our amino group, and then we have our R group. So this amino acid, remember, all amino acids will have an amino group, a carboxylic acid group, a hydrogen, and another thing attached, a side chain called an R group. So this one also has the carboxylic acid group, the amino group, the hydrogen, and the side chain, which in this case is a methyl group CH3. And the name of this is called alanine. Now, if we're talking about this isomer of alanine, it's called L-alanine, and it's referred to as the left-handed alanine. D-alanine is referenced as the right-handed alanine, strangely enough. So here is another mirror image of alanine. And we have our right-handed on this side, which makes a little more sense to me since it's on the right hand of the screen and our left-handed on this side. One thing that you may notice different about this image compared to this image is if we look at this, the carboxylic acid group, the hydrogen is there, and hydrogen here attached to the nitrogen, there are only 2 of them. In this case, we see that the hydrogen is gone, and in actuality, where it went was to that nitrogen. So atoms, amino acids in solution take on this characteristic, and the hydrogen from the carboxylic acid group can be transferred to the nitrogen, which results in this region having a slightly negative charge and this region having a slightly positive charge. Now that concept, that form of the amino acid is called a Zwitter ion. I'm writing with a mouse right now, so it looks like I'm a kindergartner. So it's called a Zwitter ion. So this is the Zwitter ion form of the left-handed alanine. So another thing that I want to emphasize, in order to have a chiral molecule, you need to have 4 atoms, or 4 different atoms or groups attached. So you can see this chirality. We have hydrogen, bromine, iodine, and chlorine attached to this chiral carbon. So they will behave differently than each other, even though they have the same atoms attached or groups attached. They are isomers of one another and often take on different characteristics than each other. So a chiral isomer is a pretty special type of isomer. Here's another reinforcement of the idea of molecules having handedness. So with these hands in the background as a guide, you can see the hands are mirror images of one another. We have our chiral carbon here on each of them. One group, two, a third group, and a fourth group. 4 different groups attached to the chiral carbon allows for the formation of chiral molecules. Molecules, isomers that are mirror images of one another. They are non-superimposable, they have different properties, and they will behave different chemically. So 19 out of the 20 natural amino acids are chiral. There is one that is not, and that is glycine. And the reason that glycine is not chiral is because you can see here it does not have 4 separate groups attached to that chiral carbon. 2 of them are the same as each other. So the R group in glycine is just a hydrogen atom. So glycine is the one amino acid that does not have stereoisomers or chirality. There are not 2 isomers of glycine that are mirror images of one another. So one of the main things I want to emphasize again is nature in the human body will only use left-handed amino acids to build its proteins. I show this in a part 2 of my protein presentation, the chirality of these amino acids. I'm going to review this again because I think it's very important. We have a central carbon here, and we have 4 things attached. We have our carboxylic acid group with the oxygen, the hydrogen, and the double bond to the oxygen. We have our amino group. We have our hydrogen right here, and this pink part is the side chain, which can be a variation of different things. And those side chains can have different characteristics, which will take us into the next part of this series of videos on proteins, which will discuss how we can classify amino acids. I twisted the molecules around a little bit there, and I will tell you that my favorite way to represent an amino acid is just like this right here. I like to have my central carbon. I like to have my carboxylic acid group on the right hand side. I like to have my amino group on the left hand side. Again, I apologize about the writing here. I feel like I'm writing like a kindergartner. And then the hydrogen and the R groups are going to be attached to the middle. So the structural formula here does not have the actual molecule does not have the hydrogen coming up like that. It's coming up in the air at you. But I like to set my amino acids up like this. And the reason for that is when we start talking about amino acids coming together to form proteins as chains of amino acids, they're often represented with this orientation, and I will continue to use it. So another little bit of review, I introduced the concept of a Zwitter ion. If I were to draw the Zwitter ion of this, I would have my carboxylic acid drawn as a carboxylate ion that's negatively charged. I would have my amino group with that extra hydrogen, and this has a positive charge. So we have charged regions on each side. And then I would have my hydrogen in R group just like they were. So this is the Zwitter ion form of that amino acid. So the next part we'll talk about classification of amino acids, and we will start with this picture right here. If you want to get a head start on it, pause this, look at it. I'm going to ask you to do the same in the next presentation, but if you do it now or you do it then, it doesn't really matter. So thanks.